Our next speaker is Dr. Howard Strickler. Dr. Howard Strickler is professor of epidemiology and population health. He completed a residency in preventive medicine at Johns Hopkins and a master's in public health at the Bloomberg School of Public Health at Hopkins before starting his research career at the National Cancer Institute of the NIH, where he was a senior clinical investigator in the Division of Cancer Epidemiology and Genetics. While at NIH, he was twice recognized with medals from the U.S. Public Health Service for his research accomplishments. He was recruited to Einstein in 1999, where the cross-cutting nature of his work has been recognized by his membership in 3 of our NIH-funded centers: the Cancer Center, the Center for AIDS Research, and the Diabetes Research and Training Center. Dr. Strickler is the author of more than 80 peer-reviewed papers, reviews for numerous journals and NIH study sections, and serves on several policymaking panels. He's a practitioner of the relatively new field of molecular epidemiology. In fact, he developed a course in molecular epidemiology, which he teaches in our Master of Science program, part of our CTSA. Dr. Strickler's work bridges the perspectives of infectious and neoplastic diseases. He's especially recognized for his research regarding human papillomavirus and cervical cancer, and most recently has been recognized by NIH for his studies of the insulin IGF-axis, research NCI named a research highlight for 2008, some of which he will discuss today. I look forward to hearing and learning more about these studies in his talk, The Insulin IGF-axis in Health and Disease. Please join me in welcoming Dr. Howard Strickler.
So I want to thank the organizers for inviting me today. It's my first time in this auditorium, and it's very nice. Could use a little artwork on the walls. So I'm going to talk about recent research from my research group, all published this year, regarding the insulin IGF-axis. I'm primarily going to focus on our research in relation to cancer and what we think is a very new area, its implications for viral pathogenesis, but and talk about the clinical implications and the road forward there, but also mention how we think these same pathways are relevant in possible other disease pathways and some of the research going on in the Department of Epidemiology to address these things. So we've got a mixed audience today, so let me just say that the IGF-axis is a critical regulator of cell growth, proliferation, and survival that affects nearly every organ system in the body. There are 2 IGFs, and IGF-1 and 2, and 6 IGF binding proteins, cleverly called the IGFBP1 to IGFBP6. So IGF-1 is a strong mitogenic and anti-apoptotic factor. Following birth, it is the IGF with the major role in regulating cell survival, proliferation, and differentiation. It mediates many of the effects of growth hormone. Most cells in the body express the IGF-1 receptor. Circulating hormonal IGF-1 levels are mainly produced in the liver. Many tissues produce IGF-1 locally, though, which is relevant as an apocrine or paracrine factor. And IGF-1 production is largely regulated by growth hormone in a negative feedback loop, but not all. And I'm foreshadowing here, not to be subtle, that IGF production in the endometrial stroma is regulated by estrogen, as an example. So this is a cow. No, this is a very big dog, and this is a little dog, and what is being shown here is just how important IGF is to growth. A single IGF, single nucleotide polymorphism haplotype is common to all small breeds and nearly absent from giant breeds. So, the IGF binding protein, IGFBP3, is the most abundant IGF in the body, and it's important to point out that IGF binding proteins are more than transport molecules. For example, IGFBP3 has independent effects opposite IGF-1. It is anti-mitotic and pro-apoptotic. It binds cell proteins in the nucleus, suggesting a direct role in the modulation of gene transcription. You can tell I got this from a junior faculty member because I don't know how to turn off the repeat sign on that. So this is just to remind me to tell you that insulin shares 40% sequence homology with IGF-1, as well as mitogenic and anti-apoptotic activity and downstream signaling pathway through the insulin receptor substrates. So let me get right into a discussion of our data in relation to cancer. Starting with laboratory data that existed before we began our work. IGF-1, we talked about being mitogenic anti-apoptotic. IGF-1 levels and its receptor expression are elevated in certain cancers. IGF-1 receptor activity is important in the establishment and maintenance of the transformed phenotype in vitro. Downregulation of this receptor results in apoptosis and growth inhibition of cancer cells. And similarly, related to insulin, similar effects in vitro in terms of being mitogenic and anti-apoptotic. Its expression, it's overexpressed and leads to transformation of cells. High insulin causes aberrant tissue development. Insulin receptors are highly expressed in a variety of cancers. We were particularly interested in studying several cancers that are common and have been shown to be associated with obesity and diabetes. And we focused on colon, breast, and endometrial cancer. And what I can say is, despite these relationships with obesity as well as diabetes with these common cancers, epidemiologic studies of insulin IGF-1 have given conflicting results. And here it's very important to be doing prospective cohort studies so that you are looking forward in time. So when the samples you have today, are they predictive of disease later? Cross-sectional studies, which are very common in clinical research, you can have reverse causality. The disease process itself could be affecting the process. So it's important to do these large, very expensive, unfortunately, prospective cohort studies. And few studies measured insulin directly, largely because they lacked fasting specimens. And the problem with that is they often ended up measuring C-peptide, which is a measure of insulin secretion. It's a more stable protein, but it also changes postprandially. So it's not a particularly excellent marker to use that in studies like these large prospective cohorts when you lack fasting blood specimens. And even when you have fasting specimens, you're not actually measuring the thing itself, which you think is involved, which is insulin. Also, no prospective studies had assessed free IGF-1, which is the putative bioactive agent, and only one cohort study had measured estradiol, though estrogen levels, like insulin, are correlated with obesity, associated with cancer, and sex hormones and insulin IGF-axis interact. The one cohort that did lacked fasting blood and measured C-peptide instead of insulin. Now, I'm going to jump straight into our data in order to save time. I just want to point out that one of the reasons we were so interested in studying estradiol in this connection is when you have high insulin levels, you have decreased sex hormone binding globulin, increased estrogen receptor expression and binding capacity, decreased IGFBP1 levels, increased free IGF-1. Estradiol, in turn, increases IGF-1 receptor expression, decreases IGF binding proteins, IGF-1, increases estrogen receptors responsiveness, and the conversion of estrone S to its active form, estrone. So the relevance of controlling for all of these factors concurrently was something that we really felt we needed to contribute in this new set of research. So we formed a collaboration with the Women's Health Initiative. They had fasting bloods. It's a large prospective study of almost 100,000 women. The mean follow-up in our study was 77 months, only 2% lost to follow-up, 5% deceased. They obtained risk factor data, annual questionnaires to obtain self-reporting clinical events. And here are the cancers that we studied. We had 300 endometrial cancers, 900 breast cancers, 500 colorectal cancers, all of which occurred incidentally during follow-up. And we used a single representative sub-cohort in what's called a case-cohort study design as the comparison group. And we measured total IGF, free IGF-1, IGFBP3, fasting insulin, glucose, and estradiol. And I want to state in advance what our a priori hypotheses were, not all of which turned out exactly as we predicted, but in interesting ways, many of the predictions were correct. So We expected that total IGF-1, free IGF-1, would be positively associated with endometrial cancer with a stronger association with free IGF-1, an inverse association with IGF-BP3, and a positive association with insulin and estradiol. For breast cancer, we expected that we would see effects with insulin and estradiol. It was less clear regarding total IGF-1 since studies had shown, interestingly, that postmenopausal cancer was not associated with IGF-1, whereas premenopausal cancer was, and conversely, that insulin and obesity is high risk for postmenopausal cancer, but it's actually possibly even protective against premenopausal cancer. So an interesting history there, which I'd be interested to hear comments on, because I've not heard any good explanations for that divide. So jumping into our data, here's results from the study of breast cancer. You can see this large insulin effect, a highly significant estradiol effect. I'll come back to this effect. And here's data on BMI. It's a little complex. All of these models, all of these analyses were adjusted for age, race, ethnicity, smoking, parity, age at menarche, age at first birth, use of OCs or contraceptives, NSAIDs, physical activity, family history of breast cancer, alcohol consumption, and education. And we see this power effect even after controlling, this power effect with insulin, even after controlling for all of these other factors here in the model. We also see a strong effect related to estradiol. So insulin is showing itself to be independent of estradiol. And what's interesting here, as it relates to BMI, when you control for all of these factors, BMI is still an important risk factor. Less so perhaps when you control for estradiol, but when you control for insulin, the effect becomes non-significant, suggesting that the insulin may be playing a major role in explaining the BMI effect. For endometrial cancer, and here we're focusing on endometrioid adenocarcinoma, we see our first set of surprising results, which is, first of all, that there's this interaction by BMI. So less of an effect of our factors. Here's free IGF-1, here's insulin. Bigger effect among those with high BMI with a very strong insulin effect. This is controlling for estradiol and other factors. And here is an inverse association with free IGF-1. And at first we didn't know what to think of this because we had predicted, as I said, a positive association. But when we went back to the literature, we found that in fact almost all of the prior studies, cross-sectional in nature, showed this same inverse association, but none of them had made much of the story. And this is why I foreshadowed that issue over the fact that there's a disconnect between IGF in serum and IGF levels in the endometrium. In fact, IGF levels in the endometrium are controlled by estrogen and are a big factor in the normal menstrual period. And so we think that there's disconnect between those levels for this particular tissue. And in fact, it's known that in serum, there's an inverse correlation between IGF-1 levels and inflammatory cytokines. So we're thinking that that could potentially be the association there. We see this strong effect of insulin now, independent of the effects of estradiol. But we don't see these effects as much here, which is interesting. Other studies have also shown this interaction. So we think that there's other factors involved with being obese that are necessary to fully express the effects of insulin in this relation. Here's the colorectal data. And this is a busy slide, so I'll just walk you through it. Here's waist circumference, here's insulin, and here's free IGF-1. Again, we've controlled in all these models for age, ethnicity, smoking, use of non-steroidals, physical activity, family history of colorectal cancer, and alcohol consumption. And the reason I put up this busy slide is you can see after adjusting for that, All of these factors are significant. Waist circumference, insulin, free IGF-1. But when you control for each other, they kill each other's effects. So I'll come back to that in a second. So it suggests either they're part of a single pathway and we're over-adjusting, or that in fact they're nothing more than surrogates for another factor which we haven't yet measured. What was most surprising, though, is we saw a positive relationship between estradiol adjusting for everything, including all of these other factors, with risk of colorectal cancer. And again, these are prospective cohort studies. So we went back to the literature because, in fact, hormone replacement therapy is known to be protective. So this was something of a surprising effect. We didn't thought estradiol would perhaps even would turn out to possibly be protective. But when we looked at the in vitro data, much of that data suggested that estradiol had a proliferative effect on the colon. And in fact, oral estrogens have a major first pass effect. In other words, a large bolus of ingested estrogen has a big effect on protein synthesis in the liver and has a profound effect on a number of different factors. And so what we're interested in now is to see effects in transdermal use of hormone replacement therapy, which hasn't been researched yet. And we're following up on this in a new set of studies that just got funded by NIH. So just to summarize these data, insulin may help mediate the relationship of obesity with breast cancer and with endometrioid adenocarcinoma of the uterus. Free IGF-1 may be inversely associated with endometrioid adenocarcinoma. And for colorectal cancer, we saw at least two independent pathways related to colorectal cancer. One that involves endogenous estradiol, and a second, what I would say is broadly associated with obesity and insulin and free IGF-1. And here I would think that we're probably, these are acting as surrogates rather than the factors themselves. But all these things offer opportunities for intervention. In particular, bringing down insulin levels is something that's being talked about using metformin and so on. And there may be other opportunities for reducing insulin levels, affecting these pathways. And we've already been speaking with Dr. Pessins from the Diabetes Center about different opportunities here to work with the basic scientists to identify different procedures. But we could even be using animal models now to see if these available insulin sensitizing drugs that would bring down insulin levels could be used in chemo prevention. So I'm going to jump forward now to some of our data in relation to viruses. These same mitogenic anti-apoptotic effects of IGF-1, presumably through effects on replication and keeping cells alive, could affect other processes. And in particular, I'll start with HIV. We thought that IGF-1, high IGF-1 would be protective because it would lead to increased survival of cells and replication in the stem cells and survival of immune cells in the periphery. So we conducted a large longitudinal cohort analysis based in the Women's Interagency HIV study. Large study of both HIV positive and negative women. There's semi-annual clinical visits that include CD4 T cell count, HIV RNA levels. We measured baseline IGF-1 and IGFBP3 in a large subset of these patients, and we looked at the pre-heart era. We focused on the pre-heart era because we, in this large pilot study, were only using baseline factors, but we were also interested in looking at the biology here unaffected by heart, because it's known to have big metabolic effects, and we would need to have repeated observations over time. So we saw an inverse association of IGF with age, which is a well-known association. IGF-1 levels go down with age. Some suggestion of decreased levels with race. IGF-BP3 levels increased with IGF-1 levels because their production is correlated. And here you see cross-sectionally, these first initial data are cross-sectional, a strong association of IGF-1 levels with CD4 cell counts in these HIV-positive patients. A significant inverse association of high IGF levels with HIV RNA. And for hepatitis C, we saw decreased levels since IGF is produced in the liver. For IGF-BP3, the same sort of age effect, some suggestion of a racial effect. Hispanics, although here the difference was the black population was also a little bit low. Again, the correlation with IGF-1, and exactly as predicted, the opposite, that high IGF-BP3 levels were associated with low CD4, and that high levels were associated with high HIV RNA levels, and again with hepatitis. But that's cross-sectional data. Here's the prospective data. Looking forward over time, what we showed is a nearly threefold increase in risk of AIDS among HIV-infected women who had the highest quartile of IGF-BP3 results. We saw the predicted inverse protective association with high IGF-1 levels, although this didn't lead to significant results. But here, when we looked at rapid CD4, both were significant, again in the same predicted direction. High IGF-BP3 was bad, low IGF-1 was helpful. And again, the implications to the treatment of HIV are hopefully reasonably obvious. These are hormonal pathways that we can manipulate, and we've just received some new funding to continue this research. And we're going to start also doing some research into the polymorphisms. So what about hepatitis C? So most circulating IGF-1 and IGF-BP3 is produced in the liver. IGF-1 stimulates proliferation, collagen gene expression, and accumulation of hepatic stellate cells, the primary source of extracellular matrix and liver fibrosis. IGF-1 promotes chemotaxis of these cells to liver injury, whereas IGF-BP3 does the opposite. So here, just like in cancer, and unlike in HIV, we thought that IGF-1 would be harmful because it would lead to the proliferation and collagen gene expression of these cells, and IGF-BP3 would be helpful. And that's exactly what we saw. A highly significant protective effect using APRI elevation as a specific but non-sensitive measure. Looking forward in women who started off with normal levels, developed evidence of APRI elevation forward in time, suggesting that they had developed some degree of liver disease, and IGF-BP3 levels were highly predictive of those events, and IGF-1, I mean, it was an extremely strong effect, nearly six-fold increased risk. I'm sure these levels are exaggerated. I don't think that the effects are going to turn out to be that strong, but we're already moving forward on new projects. And this will be my next to last slide. So we looked also at human papillomavirus, and again, we expected making cells replicate and keeping them from dying would be bad for HPV. And indeed, in this very small pilot study, only about 150 women, we showed that IGF-BP3 was inversely associated with the risk of incident oncogenic HPV, and the IGF-1, IGF-BP3 ratio was positively associated. In terms of clearance, high IGF-1, low IGF-BP3 was associated with an inverse association suggesting increased persistence. So high IGF-1, low IGF-BP3 led to greater persistence of the HPV, and increased, and IGF-BP3 was protective against SIL. Those are the significant results, but overall, in the small pilot, the data, while not everything was exactly consistent, it's small pilot, and what we see is that overall, high IGF-1 is associated with risk of HPV, IGF-BP3 protective. And again, we see real opportunities for manipulating these factors for the sake of development of new therapies, and we're moving forward quickly to try to move from these studies to clinical trials. And I'll just mention, in our department, recent papers coming out from the work of Dr. Robert Kaplan showing high circulating IGF-BP1 levels may be associated with congestive heart failure in older adults. High IGF-BP1 levels were associated with greater risk of mortality and poorer functional ability. With diabetes, high IGF-BP3 and low IGF-BP1 were associated with insulin resistance. IGF-1, just like insulin has IGF-1-like effects, IGF-1 has certain insulin-like effects. So this is some of what's going on in our department, and we look forward to working with our laboratory and clinical investigators to quickly move these results towards their clinical use. So thank you for your attention.